Hey everyone, welcome to Professor Long's lectures in microbiology. I'm Professor Bob Long. If you've been watching my videos, you know that these videos are intended for use by students who are enrolled in my course at Del Mar College. It's microbiology for the health sciences. If you are not enrolled in my class, please know that your instructor may go into more detail, explain things differently, and want you to learn slightly differently from what I do. I'm trying to provide a basic fundamental approach to the foundational principles in microbiology so that my students can begin to add detail as time goes on. I want you to be able to open a textbook and read it and understand what it's saying when you get into the next phase of your program, what we call the professional phase in nursing school, medical lab tech, surgical tech, or whatnot. So, if you find the videos helpful, please hit like and leave me a note so that I know to keep doing them. And one last thing, as I like to tell you guys in the beginning of these videos, these are done very crudely. It's a cell phone, me, and a board. We are in a short time crunch because of the COVID situation, so I'm pumping these videos out rapidly. As I learn the technology, I hope to clean these up and make them much nicer, much more edited, and more professionally done. All right, so what we have been talking about is the immune system. We've already talked about bacteria and viruses and other pathogens, helminths, and things. What we're trying to get into is the application of all this stuff. Why is it important for health science majors? What do we need to know? Well, we've begun talking about the immune system, our body's defenses against foreign invaders. Those foreign invaders can be bacteria, viruses, eukaryotic cells that are pathogenic to us. Whatever foreign invader can cause an infection, even exotoxins. Okay. Now, when we first began talking about the immune system, we were talking about innate immunity. So I want to start off by differentiating between two parts of our immune system. Our immune system can be divided basically into two different subsystems. There's innate immunity, and as we said before, innate simply means inborn immunity. So a few details that I want you to know about this immunity, okay? You are born with it, okay? As you enter this world, these cells are already in your body. They provide a rapid response to a pathogen. That rapid response is usually a local response. It's at the site of infection. So if you get a cut in your skin and some bacteria gets in, then you have some white blood cells, and certain ones, which we'll name in a minute, we've named them before, that will immediately attack it in that location, and that's about it. One of the things that we know is that it involves phagocytosis, okay? Or lysis of the foreign cell. Somehow, some way, our white blood cells will kill this foreign cell. They either engulf it by phagocytosis and fuse it with lysosomes, which have digestive enzymes and digest it, or sometimes they can release some cytotoxic chemicals that will kill the foreign cell. And the cells involved include macrophages. Macrophages come from a class of cells called monocytes. So it's monocytes and macrophages. And there's a cousin of these called dendritic cells that we haven't really talked a whole lot about. In addition to macrophages, it involves neutrophils. It involves eosinophils. And it involves basophils. Okay? There's actually one type of cell that's related to a lymphocyte called a natural killer cell. They are derived from lymphocytes. All of these cells are either phagocytic or involved in releasing cytotoxic enzymes that can kill the foreign cell. They also trigger the immune response the immune response. They trigger the inflammation response or inflammatory response. And in inflammation, blood vessels dilate, become more leaky, fluid leaks out into the tissue. That tissue becomes swollen and red and tender, and these cells can more easily migrate out of the bloodstream. We did talk about chemotaxis and diapedesis, their movement out of the bloodstream into the peripheral tissues, and they will directly attack the foreign invader. We call that innate immunity. Innate immunity, by the way, is also called the non-specific immunity. Okay? You're born with it, innate, inborn. It's non-specific. It doesn't matter what the invader is, these cells will attack it and wipe it out. Okay? The opposite of that is the one that we want to talk about today, which is goes by 3 different names. It's called acquired immunity, it's called adaptive immunity, and it used to be called specific immune system. My penmanship's not so hot right now, but yeah, I've had that problem for a while. So let me adjust my camera just a little bit. So when it comes to acquired immunity, adaptive immunity, or what we call specific immunity, and let me rewrite that. That's a little bit messy, and my marker is starting to get a little bit faint. But in what is called acquired immunity, also called adaptive or specific immunity. Now, it's called acquired or adaptive immunity because we don't have it when we're born. We have cells that are capable of the responses, but they haven't been activated yet. And the reason they're not activated is because they've never seen or been exposed to that foreign invader, that pathogen, or some toxin. And so our body has to adapt to this new situation, something that it's never seen before. And we acquire it only after exposure. And it develops so that those cells that are involved attack that specific invader. And they can remember it. So one of the things that we talk about, one of the things I didn't mention under innate immunity is that there's no memory component. These cells don't need to remember it. They just attack whatever it comes into the body. Now, this is sort of a real general rapid attack. Kind of like just having a security force cruising the body. Something comes in, they try to attack it. Sometimes they alert a few other cells. But when the general immune response, when our innate immunity and non-specific immunity doesn't function in completely wiping out the invader, if the invader can get into the body and cause an illness and a disease, then we activate the acquired immune system. Okay, so the acquired immune system is kind of like a small fighting unit that goes in to attack a specific group, a specific invader, and not worry about all the other areas. Now, a few details about acquired immune system. You are not born with it. You're born with the cells, but they are not activated and ready to fight specific infections. It is a slower response, at least under what we call the first exposure. I'll explain that a little bit later. The first time, well, I'll explain it now. When something invades your body, these guys got to scratch their head and figure out, what is this? We know it's not part of our own body. It's a foreign cell. How do we attack it? And it takes a little while. And then they develop the antibodies in the system to attack it. They can wipe it out. And it's a really strong attack. But some of the cells hang around and remember it. So even if some are killed off or die in the process, some hang around. The second time this invader tries to come in the body, they are already capable of recognizing it. So on second exposure, you get a much more rapid response. But the initial exposure gives us a slower response. Does not usually involve phagocytosis, at least not initially by these cells. Now, what we're going to learn is under the acquired immune system, once it's activated, it can alert some of these cells to come rushing in and phagocytose what we call antibody-labeled cells. But the cells involved in this don't usually themselves phagocytose things. We're going to call these cells lymphocytes, and they're usually not involved in that. It does involve phagocytosis to some degree, but the lymphocytes that are involved, the T cells and B cells, do not personally engulf the foreign invader. Okay. Something else I want to does involve the release of antibodies. We're going to talk about antibodies. They're proteins that are released that can bind to foreign cells. They recognize them and bind to them, and it sort of is a way of saying, hey, come get these guys. So imagine if you have a big military, a big group of bad guys that can run around, or good guys I should say, that can run around and take out the bad guys. But sometimes they don't know who the bad guys are. So in our acquired immune system, sometimes our cells can tag the bad guys. It would be like hitting them with paintballs of a certain color, so that now the other cells can move in and wipe out the cells that are kind of labeled with these antibodies. It's sort of like a molecular tag that says, hey, here, I'm a bad guy, and we can wipe it out. The cells involve usually what we call lymphocytes. We've talked about all these white blood cells. And to some degree what we call monocytes and dendritic cells. Okay? Macrophages are dendritic cells. They are part of the innate immune system, but they can play a role in activating the acquired immune system. And we're about to see how. Okay? So, I want you to know some of the differences. Innate immunity is one that you are born with. Innate means inborn. It's non-specific immunity. It has a very rapid response that is local. There's no memory component. Most of the cells will come in and simply attack other cells and phagocytose them or lyse them open with some cytotoxic enzymes. It involves macrophages who can become it involves monocytes, which are called macrophages if they live out in the peripheral tissues. And dendritic cells are just cells that are kind of stuck out in peripheral tissues, as well as mast cells. It involves neutrophils, eosinophils, basophils, and some natural killer cells that are related to lymphocytes. All of this is part of the innate immune video, so if you want to watch that in review, you can. The acquired or adaptive or specific immune system, which is the focus for today, you're not born with it. It's a much slower response, at least upon initial exposure. It does not usually involve phagocytosis, usually not by the lymphocytes, and does involve the release of antibodies against a specific invader. Those antibodies will label it so that other cells can engulf it or kill it. It can involve macrophages to some degree, but remember, macrophages are not part of the acquired immune system, but they can trigger the acquired immune system. So how does all that work? Okay, I'm going to try to draw a simplistic series of pictures that try to explain this in a very simplistic fashion. There's a lot of different ways we could go about explaining this, and we could get lost in the weeds talking about all these other chemicals and how they play a role. I'm really not interested in that right now. So, essentially, let's say a foreign cell, like a bacterium, or not even necessarily a cell, maybe a virus. But here's our little bacterium. This bacterium has on its surface a bunch of proteins, and there can be different proteins. I'm going to give them different shapes. Those proteins on the surface of a foreign cell are called antigens. Okay, they're called antigens because they generate, that's the gen part, an antibody response. That's the anti. Okay, they are antibody generating. So they're called antigens. And I've written where you can't really see. So let me raise my screen just a hair. Okay. So now, we have a foreign cell with surface antigens. By the way, our cells have markers on them as well that can be called antigens. And they are markers that tell our immune cells, my cells belong to me. And as our immune cells cruise around and check our surface, the surface of other cells, If those cells have the correct markers, they leave them alone. But when they have some kind of foreign marker that they don't recognize, the cells of the innate immune system will simply engulf this cell. And that's kind of what happens. So initially, what we're going to talk about is a cell called a macrophage. So macrophages are derived from monocytes, and sometimes we call them Big Macs because they're huge compared to other cells. And that macrophage will engulf this bacterium or foreign cell, and essentially what happens is, as it takes it in, it phagocytoses it into a vesicle. So my little bacterium is here with these surface antigens on it. Okay. Now inside the macrophage, there are bubbles of membrane, vesicles called lysosomes, and the lysosomes will have enzymes in them that when they fuse with this vesicle, they will release those enzymes, and the enzymes will digest the bacterium into a bunch of little pieces. Now there's another molecule inside of here that are called MHC molecules. I'm going to draw one here. And these MHC molecules can recognize and bind to a specific surface antigen. And as the macrophage does that, as it has digested this, and these MHC molecules can enter into these vesicles. Inside the vesicle, I'm going to blow this vesicle up a little bit. Inside the vesicle, the MHC molecule will have a specific three-dimensional shape that allows it to bind to this surface antigen from the bacterium. Now, because that's inside this vesicle, when this vesicle fuses with the cell to try to release any of the debris that was digested, this MHC molecule remains in the membrane because it becomes bound to the membrane of the cell, and it will have on it our specific antigen. Okay? So when a macrophage engulfs a bacterium or some foreign invader, it will digest it. There's a molecule in here called an MHC molecule. MHC stands for Major Histocompatibility, there's a word, complex. Okay? It's a complex, a group of molecules that can be compatible histologically with certain foreign cells. And so they're called the Major Histocompatibility Complex. And there's MHC1s and MHC2s. I'm not going to get too much into the detail, but suffice it to say that this is an MHC2 molecule for now, okay? Since we've engulfed a foreign cell. MHC1 molecules would be generated if the foreign invader was already inside the cell and we digested it like viruses. Then we attach an MHC1 molecule to it. And then that would be on the surface of our cell. But nonetheless, so this MHC2 molecule is here, and the little tan molecule is called the antigen. And I will usually abbreviate it as AG. That antigen has a specific amino acid sequence. They're usually proteins. It has a very specific amino acid sequence that matches specifically with a specific MHC molecule, and then they get presented on the surface. And as it engulfs many of these, we can get these MHC molecules all over the surface of our macrophage now, and they will all have this little antigen bound to it. I'm drawing the antigen like a little diamond for a reason, okay? Now, once this macrophage has the MHC molecule and the antigen, now I'm going to erase this, but all of that happens, the binding of the MHC molecule happens inside the lytic vesicle, and then it's presented onto the cell membrane. Once this is done, we call this a professional antigen presenting cell. Some people use the term PAP-C, and that PAP-C stands for Professional Antigen Presenting Cell. These guys are pros. Anyway, it's an antigen presenting cell. It's going to present this antigen to other white blood cells. So I'm going to erase this part right here. I'm going to erase this part because we already got it down. So now what happens? Our macrophage has engulfed a foreign cell, digested it, bound some antigen from that foreign cell to a specific MHC2 molecule that is specific to that antigen, and then presented the antigen on its external surface. Now there's another type of white blood cell that we did not talk about before in great detail. This white blood cell is called a lymphocyte. There's different types of lymphocytes. Okay? Now lymphocytes are pretty small white blood cells. Now these lymphocytes can have on their surface, they can come in different types. There's T lymphocytes and there's B lymphocytes. T comes from the term thymus. That's where these lymphocytes mature in the thymus gland. And some are called B lymphocytes for bone marrow. Actually, it's the bursa fabricius, but we're not talking about it. So B lymphocytes mature in the bone marrow. T lymphocytes mature in the thymus. That's where they kind of get their name from. There's a specific type of T lymphocyte called a T helper cell. So T helper cells, I'm going to abbreviate as a T with a little H on them. T helper cells have on their surface another protein that might recognize this specific antigen. So, since this specific antigen has a specific shape, if a T helper cell has a surface antigen, a surface molecule that can bind that surface antigen, it will bind to this antigen presenting cell. And by the way, the protein on the surface of a T helper cell is called the CD4 molecule. It stands for I'm drawing a blank on the CD, but anyway, anyway, it'll come to me in a second. So the CD4 molecule is unique to T helper cells. Now each T helper cell can make only one type of CD4 molecule that can recognize only one specific antigen. But I can have millions of different CD4 molecules. If the correct CD4 molecule encounters this antigen as it's being presented, then that CD4 molecule becomes activated. And one of the things that happens in the activation is this macrophage can release a molecule called interleukin 1. And we will abbreviate it as IL-1 sometimes, okay? So, interleukin 1 is released by the macrophage, or a dendritic cell in some instances, and that will stimulate or activate this CD4 molecule as it's bound here. That causes this CD4 molecule to release another molecule called interleukin 2, or IL-2. Now, inter means between, and leuco means white blood cells. It communicates between white blood cells. As this T helper cell is activated, one of the things it can do, in addition to releasing interleukin 2, is that this cell will start to divide by mitosis, and I will get a whole lot more of these CD4 cells. And because their genetic makeup is for this specific CD4 molecule, the CD4 helper cell or the T helper cell, I can get tons of these T helper cells that are now all going to have these CD4 molecules that are specific for a specific antigen. Okay? So now they can go bind to other cells that are presenting antigens and multiply and multiply and multiply, and then they can release tons of these interleukin molecules. They will flood our body with interleukin molecules. Those interleukin molecules can enter your bloodstream and travel all over and attract other cells. These interleukins, by the way, belong to a class of molecules called cytokines. Okay, these cytokines they're numerous, but a couple of them are interleukin 1 and interleukin 2. Now, one of the things, or there's 3 things that these activated cells can do. So I'm going to erase a little bit of this. Okay, as they release the cytokines, one of the things that can do is they can alert the innate immune cells. So what they can do is they can basically tell the innate immune cells, hey, listen, we have an infection here. You guys better hurry over. And so what happens is I can get more MACs, more neutrophils, more eosinophils, and more basophils and natural killer cells to come in and start wiping out these guys. So they can alert the innate immune system, hey, there's an infection, come on now. And they will multiply by mitosis big time and increase the size of our army. Plus, they trigger what we call the inflammation or the inflammatory response. Note, you can recall that basophils can release histamine, and histamine can dilate blood vessels and cause leaking into the blood vessels, and we get all the inflammation. If you don't know what all of that is involved, you need to go back and watch the last video. So that's one of the things that these activated CD4 or T helper cells can do. A second thing that the T helper cells can do is they can activate what are called cytotoxic T cells or T killer cells. So I'm going to make these guys in red because they're bad boys. So a second thing they can do is activate cytotoxic also called killer T cells. Come on, Bob. Okay, so these cytotoxic T cells have in their membrane another molecule that can also match these antigens. And what they can do is, and that molecule, by the way, is called CD8. So sometimes these are called CD8 cells. Cytotoxic T cells, killer T cells, or T killer cells, and T cytotoxic cells, our CD8 cells are all the same thing. Now what happens is these T cells have on their surface these CD8 molecules that can recognize the exact same foreign antigen that was on the original invader. And they will multiply rapidly by mitosis and increase in number. And since this specific T cell, cytotoxic T cell, is being stimulated, then they will all have the exact same CD8 molecule that can recognize just that specific surface antigen. So now, let's say I have a bacterium that comes along and it has the correct surface antigen on its surface. Then this CD8 molecule, or CD8 cell, or T killer cell can bind to it, and it will release molecules called perforins. And those perforins are like little daggers, and they can go and stab holes in the invader and kill it. Okay, let me make sure you can still see where I'm writing on the board. I don't need to move my camera, okay? No, you can still see. So now, the T helper cells help the whole process by releasing these interleukin 2s or cytokines, and those cytokines can stimulate the innate immune system to increase inflammation and send a massive army over, but they also can stimulate the cytotoxic T cells. These cytotoxic T cells will bind to only that specific foreign invader. If I had a different bacterium nearby, and let's say it has a different shaped antigens on its surface, then this CD8 cell or cytotoxic T cell will not recognize it, nor would this T helper cell, because these surface antigens, this CD4 molecule and this CD8 molecule, will be specific for the surface antigens of a specific invader and not others. Okay? So these other guys would get by unless they get engulfed by a macrophage, and we set off a whole second class of lymphocytes, T helper cells, cytotoxic T cells, that are now specific to that invader. This is why this is called the specific immune system. It's also called the acquired immune system, because we have to acquire these cells. They have to adapt to that specific invader. Every time a new foreign invader enters the body, some bacterium or virus, our specific immune system can do this. And every time it happens, those immune cells will only respond to that specific invader, and a different one will not be recognized. Okay? Now, one last thing that I want to mention before I move on to the third thing that these cells can do is that some of these cytotoxic T cells are going to hang around. I'm going to put a TCM. That's not really what we do, but I'm doing that. Some of them are called memory cells, because these memory cells will not necessarily release these cytotoxic enzymes, but if some of these guys die off after the fight, these memory cells will stick around. And if this same invader tries to invade your body the second time, they will recognize it right away, as will these T helper cells, and because there will be some memory cells that are sticking around, and those memory cells will remember this invader, bind to it, and wipe it out before it can make you sick a second time, usually. Okay? Now, I said there's three things they can do. One is it can alert the innate immune system and amplify it and inflammation to get a response. Two, it can activate cytotoxic T cells that can go in and release chemicals called perforins. They bind to the foreign invader. It's kind of like grabbing onto a bad guy and stabbing him to death. They release chemicals called perforins that perforate or poke holes in the foreign invaders and lyse them. The third thing that can be done by these T helper cells is that they can activate what we call B cells or B lymphocytes. These are all lymphocytes: T helper cells, T helper lymphocytes, T killer lymphocytes or cytotoxic T cells, and then there's B cells. Now the B cells come from, they mature in the bone marrow, and usually these cells are called naive cells. I'm going to use this term, naive. Okay? Naive means you don't know. And naive cells are cells that don't know how to respond because they've never been exposed to this specific foreign invader. But once exposed, then they become what we call competent cells. They're competent at recognizing and dealing with the foreign invader. So these B cells are naive. They're not exposed. Some people call them virgin B cells, but they're usually called naive B cells. Now what happens is these T helper cells can activate the B cells. Now what B cells do is this. When I have a B cell here with its little nucleus, B cells have on their surface these proteins called antibodies. We're going to have a whole nother video where we talk about antibodies. But antibodies are these little Y-shaped proteins. One part is called the constant region because that amino acid sequence never changes, and it allows them to bind to the surface of the B cells. On the outside, on the outer edge, there's these two spots called the variable regions because it can vary. And if I have a B lymphocyte that makes a specific antibody on its surface, then that variable region is locked in and will not vary for that B cell, but a different B cell will have a different amino acid sequence in this outer edge of this antibody on its surface. And it turns out that this antibody will be specific for that foreign antigen. It's amazing how our body can do this, and we don't have time to go into it. But essentially what happens is once a B cell gets activated, the B cell will start to divide rapidly, and some of them are going to differentiate. So we get massive mitosis, and this cell will make more of itself with that specific antibody on its surface, and it will rapidly divide. Some of them become what we call plasma cells. A plasma cell is simply an activated B cell, and what happens is if they bind that foreign antigen and I'm going to change the color of it, but that little diamond-shaped thingy from the other one, they can actually take it in, they can make antibodies, they'll make tons of these antibodies internally. They do massive protein synthesis, and then they release those antibodies to be floating out into our cytosol and into our bloodstream. Okay? So once all these plasma cells, these B cells get activated and they divide rapidly, some of them will continue to divide and become plasma cells. And all of these thousands and thousands of plasma cells will secrete antibodies that will bind to that specific antigen. Okay? And then another group will hang around and they'll have that same antibody in their cell membrane, and we call those B memory cells because they will remember this infection. Let me see if I'm writing off of my screen yet. No, I'm still on the screen. Great. The B memory cells, like the T memory cells, stick around and remember that specific foreign invader that kicked this whole process off. And if that specific foreign invader gets wiped out but tries to re-enter the body on the second time, these memory cells will recognize it, massively divide, some will differentiate into plasma cells, and they'll release antibodies against that specific foreign invader. Now, one last detail, and I'm running out of room, so I'm going to have to erase some of this, but let me reiterate something here. When a foreign invader, a specific invader, let's say it's a bacterium like Proteus mirabilis, enters the body, if a bunch of them get in, our macrophages, part of the innate immune system, and some dendritic cells and other cells can phagocytose them. As they phagocytose them and digest them with lysosomes, there's another protein inside called the major histocompatibility protein, or MHC, major histocompatibility complex. The MHC2 molecule will bind to a specific antigen, a specific protein from that bacterium, and then stick the MHC molecule with the antigen off on its surface, basically running around telling everybody, look, here I'm presenting to you the bad guy. And when a T helper cell that has a CD4 molecule on its surface that will bind to that same specific antigen, when they communicate with each other, the macrophage release interleukin-1, that tells the T helper cell to release interleukin-2, and the T helper cell will become activated and it will start to divide rapidly. And all of its cells, all of its offspring, have the exact same CD4 molecule against that exact same invader. And if a different bacterium like E. coli came swimming by, then these guys would not recognize him because he might have a different surface protein. Or if the flu virus floated by, they wouldn't recognize it. They can only recognize that specific antigen. Now, once the T helper cells are activated, they can release the cytokines, the interleukin-2s, and those can alert our innate immune cells, and they can come in, have an inflammatory response, flood the site with all these guys that can try to wipe out whatever's causing the infection. But some of them will activate another group of T cells called T killer cells, or cytotoxic T cells, also called CD8 cells because the protein in their membrane is CD8. And those cytotoxic T cells will bind to the foreign pathogen and start poking holes in it with proteins called perforins. Some of them will stick around and not get involved in the fight, but will be called T memory cells. And if that invader comes back into the body a second time, these memory cells can divide into cytotoxic T cells and wipe out the infection again and make some more T memory cells. This is why sometimes if you get exposed to something a lot, you get a stronger immune system. We're going to talk about vaccines in the future. The third thing can happen is these helper cells can activate B cells. B cells will divide rapidly by mitosis. Certain B cells have antibodies in their cell membrane that also recognize the exact same foreign antigen. Once they are activated, they will divide rapidly by mitosis. Some hang around as memory cells. They don't get involved in secreting antibodies. They just wait for a second infection or a third infection from this invader, and then they can divide rapidly. Some of those will become plasma cells. Some of them hang around as memory cells. The plasma cells will make antibodies against that specific invader and release them into the surrounding fluids. So this is going to take us to the last concept I want to talk about. If I leave out all the B cells, okay, if I just talk about this process is called cell-mediated immunity, okay? So this little process here, part 2, is called cell-mediated immunity. The reason it's called cell-mediated immunity is because it involves cells. It's going to mediate the cells and get them involved in attacking the foreign invader. The B cells do what we call humoral immunity. Humoral mediated immunity or humoral immunity means, the term humors comes from an ancient time when people thought the body had all these fluids in it called humors that determined your personality, your attitude, as well as your ability to fight off infections. They called them the humors of the body. Well, because these antibodies are released into the body fluids, the humors of the body, this is called humoral mediated immunity. Cellular mediated immunity involves the cytotoxic T cells being activated. Humoral mediated immunity or humoral immunity involves the B cells releasing antibodies into our fluids, the humors. Okay? So I hope you got a lot of that down. One last detail, and I'm going to have to erase some of this because I'm out of space. Okay? If you need to copy it all down or redo this, watch the video again, pause the video, whatever. Okay? Now when it comes to humoral mediated immunity, one of the things that happens is this. Okay? So let's say I have a B cell, or what we're going to call a plasma cell. Okay? My plasma cell has on it antibodies. These antibodies have a little area here which can bind to a specific foreign protein called an antigen. And if I have a plasma cell that is dedicated to a specific foreign antigen, then all of its antibodies on its surface are going to have the same receptor site. Okay? Now, when it binds to a foreign antigen, then it will activate this plasma cell even further, and this plasma cell will start to make antibodies through protein synthesis against that specific antigen. And it will secrete those antibodies into our humors or fluids. Okay? So now I can have millions of these antibodies against a specific protein floating around. Now we're going to go into antibodies in a little bit more detail in the future, but suffice it to say, let me knock this out real quick. Our antibodies, when I look at an actual antibody, is made up of 4 proteins that get linked together by what we call disulfide bonds. 2 of the proteins are rather large, and they're stuck together with some chemical bonds called disulfide bonds, 2 sulfur molecules. Then there's a smaller protein that is attached to this larger one here. So we call these the heavy chains because they're bigger and heavier, and these are called the light chains because they're smaller and lighter. And it turns out that most of this molecule is called the constant region. In the constant region, the amino acid sequence does not vary from one to the other. Okay? So all of these have the exact same amino acid sequence, and the part that's bound to the membrane, and in this part that will not bind to the foreign cells. But this little area right here is called the variable region. The variable region can vary from antibodies that are against a specific foreign invader. So for example, if this plasma cell is making these specific antibodies, then their variable region will be set for that particular antibody. And if I have a foreign cell that comes in and has a protein on its surface that these antibodies can recognize, then the antibodies will bind to it. Let me use the same color I was using before. these antibodies will bind to it. Now it turns out that the antibody has 2 binding sites for that surface antigen. What that means is it can actually bind to 2 of the exact same foreign cell, because these 2 foreign cells, since they are identical, they should all have the exact same surface proteins. And if I have a whole bunch of these foreign cells hanging around that all have the same surface proteins, what happens is I can actually have antibodies that bind to 2 of these. And another one here, and then this one could bind to this one, and you get the picture. Okay? So what happens is this antibody, since it has 2 binding sites, can bind to 2 of the same foreign cell. And then another one of these exact same antibodies would bind to the same antigen, because there's multiple copies of the antigen on the surface, and another of the exact same foreign cell. And another antibody can bind 2, and this one can bind 2, and this one can bind 2. And eventually we can clump or agglutinate all of these foreign cells together. So antibodies can do a number of things. Because of their variable region, this particular area will vary depending upon the plasma cell that produces it. Let's say I have plasma cell number 2. It has on its surface antibodies, but those antibodies have a different variable region. That variable region will not recognize these foreign proteins. But if it does secrete these antibodies, then whatever foreign cell has the correct antigen, then these guys can bind to it and bind up all these foreign cells. That's why it's called specific immunity. This plasma cell will make antibodies against a specific invader or pathogen. This plasma cell will make antibodies against a specific foreign invader that's different than that one. And the reason they can bind to 2 different foreign invaders is because of this variable region can be manipulated genetically, and they can alter their gene and alter the protein sequence here, the amino acid sequence of the protein. So I can have antibodies against a specific foreign invader. Now, a few things I want to mention about this. Okay, I'm going to erase this part because I'm going to need to write on the board some more. Okay. One of the things, or several things that can happen when we have this process occur. Okay. One of the things that can happen when antibodies bind is that antibodies can do the following things. One of the things they can do is they can neutralize a pathogen. They will bind up to these, let's say for example you have a virus that invades your body, and that virus wants to bind to certain cells, to membrane receptors, and then inject its DNA or its RNA genome. Well, if I have antibodies bound to that virus, the virus can't bind to the cell that it wants to. They may be on there causing problems. For example, if I have a cell that has a receptor here that a virus wants to bind to, but this virus is coated with antibodies, then it can't bind to that receptor. So they can neutralize certain cells and block the receptors or block them from binding to receptors. A second thing that they can do is they can agglutinate. To agglutinate means to clump together. So they can neutralize certain pathogens by blocking the proteins on their membranes that bind to receptors, by coating them, or they can clump them together. Now, when other cells come in to kill them, they don't have to go chasing each one down individually. They can attack a big group of them at once and wipe out a whole bunch of them. So that agglutination is to clump them together. Another thing that antibodies can do, another effect they can have, is that they can immobilize some cells. So some foreign invaders have flagella that allow them to swim around or move. And if I have antibodies binding to these flagella, it could disrupt their ability to allow these flagella to function, and so they can immobilize certain foreign invaders. So they can neutralize cells by binding to them and blocking their ability to bind to receptors on our cells. They can agglutinate them or clump them together so our immune cells can wipe a bunch of them out at once. They can immobilize some pathogens by binding to flagella and not allowing them to move around. They can activate the complement system. So we talked about the complement system last time. The complement system is a whole bunch of proteins that set off, it's over 30 proteins, that get involved in this enzyme cascade. One triggers another, triggers a couple of more, and eventually some of those complement proteins can bind to the surface of a foreign cell, and they can actually form a little pore and allow water and ions to flow in and cause lysis. So some antibodies can activate the complement system. Okay? And finally, they can play a role in oxidization. So if I have a foreign cell that's coated with antibodies like crazy, a group of foreign cells, macrophages and other white blood cells, but particularly macrophages, love to gobble up any cell that's coated with antibodies. So when we coat a cell with antibodies, macrophages will recognize them more readily and more easily and start to devour them. So not only will they agglutinate the foreign invader, they can also opsonize them, and with the inflammatory response and all the cytokines released, all of these other white blood cells will move in and attack them and help eliminate them. Okay? Now, that's a rather simplistic overview. We still haven't talked about the different types of antibodies, but the video is getting kind of long. So we're going to talk about all the immunoglobulins, IgA, IgM, IgD, IgE, next time. I wanted you guys to get a basic grasp, an understanding of how our acquired immune system works. First, know the differences. One we're born with, one we're not. One attacks just anything. The other one attacks very specific invaders based on cell surface markers called antigens. The acquired immune system involves a macrophage presenting with an MHC molecule, a surface antigen. T helper cells recognize it, become activated, multiply rapidly, and then release some cytokines. The cytokines can activate the normal immune system and increase inflammation, letting white blood cells come in. It can activate cytotoxic T cells or T killer cells, which will bind to this foreign invader and kill them. And it can activate B cells, which will begin to make antibodies. The antibodies will bind to the foreign cells, either neutralize them or agglutinate them or immobilize them or some combination thereof, activate our complement system, and opsonize them so that other white blood cells will recognize them. That's a lot of information. So I hope this video was helpful. I hope you learned something, and I hope you had as much fun as I did. If you liked the video or it was helpful, please hit like and let me know. Leave me a note. I hope to see you in the next video. We'll talk a little bit more about antibodies, the immune system, and how we can apply it with vaccines and other things. So thanks for watching.